Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to Sandesh Neupane and Derek Wright. Lentil is an important food crop grown in multiple regions around the world. However, differences in temperature and day length can limit crop breeding efforts across regions. This episode, Sandesh and Derek discuss how their research growing lentil varieties around the world and comparing their genotypic and phenotypic data can help reduce these difficulties. We'll talk more about that in a minute, but before we dive in, we want to thank our sponsor for this episode, Meter Group. Meter specializes in robust soil moisture sensing, innovative weather monitoring, cloud data logging, advanced data visualization software, and more. Their well-published scientific instrumentation is used worldwide in universities, research and testing labs, government agencies, agriculture, and industrial applications. Listen to their new podcast, We Measure the World, to hear how innovative researchers leverage environmental data to make our world a better and more sustainable place at metergroup.com/fieldlabearth. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, welcome to the show. Today we have Sandesh and Derek with us. Sandesh Neupane is a PhD candidate in plant sciences at the University of Saskatchewan, Canada. His research focuses on plant phenomics and genomics with lentils as a key crop. With a master's in plant sciences from the same institution, he conducted phenological studies on lentil germplasm worldwide as his master's thesis project. Upon completing his master's degree, Sandesh transitioned into a role as a research assistant at the same institution, where he collaborated extensively with a multidisciplinary team comprising breeders, agronomists, bioinformaticians, and computer scientists. Together, they embarked on an ambitious journey to integrate cutting-edge technological advancements, particularly image-based phenotyping, into the realm of lentil breeding. Before coming to Canada, Sandesh worked for over 5 years in Nepal's agricultural research sector, emphasizing collecting and evaluating local crop diversity. His contributions led to the registration of Ramechhap Hario Latte as Nepal's first recognized variety of amaranth species. Sandesh has co-authored about a half dozen journal articles and several conference papers related to lentil breeding. Derek Wright was born in Regina, Saskatchewan, and graduated with a bachelor's in biology from the University of Regina in 2012, followed by a master's in agrobiotechnology from University of Gießen, Germany in 2015. He now works in the plant sciences department at the University of Saskatchewan and has been involved in numerous research projects with lentil, Lens culinaris. Welcome to the show. How are you both doing today?
Very well, thanks.
Great, thanks.
Awesome. Well, we're so happy to chat with you today. Lentil is our topic if people weren't sure from those bios. But to get us started, I want to take a minute to define a couple abbreviations that might come up throughout the show. So the first one is a QTL. That's a quantitative trait loci, which is like an area of interest that might have specific genes related to certain traits in DNA. DTF is days to flowering, and LDP is lentil diversity panel. And then there's two more that I don't feel qualified to define on my own. So I will lean on Sandesh and Derek to help me define a GWAS and latent variable phenotypes.
Okay, maybe I'll go with the GWAS. GWAS is basically like we want to do the correlations between the phenotypes, like that is the observable traits that we can see, and the genotypes, which is like the genotypic informations from any varieties. And we want to see the correlations. The approach itself is called GWAS.
And then for latent variable phenotypes, this is sort of something that we kind of a little bit tried to define in our paper. It's already a bit of a concept, but we wanted to kind of take it into the kind of plant science area. So what these latent variable phenotypes are is essentially a trait that is not necessarily directly observable or easily understandable from a human perspective. So it's not necessarily something you're going to go out into the field and kind of easily measure, like say plant height or something. It's going to be more something like a a little bit more abstract, like a data point, for example, that you would extract, let's say, from drone images. That's where this kind of concept sort of came from, was extracting sort of data from imaging stuff. And so we kind of tried to broaden that concept because we ended up with a lot of traits that kind of fell into that category. Like, so for example, we had some traits that were, how we say, gave us an indication of how the temperature sensitivity of a lentil line. That would be kind of one of these latent variable phenotypes, because we can't go out into the field and easily measure how temperature sensitive a lentil variety is. However, we were able to do some kind of statistical modeling and stuff that allowed us to get that data point. So it's these kind of more abstract data points.
Yeah.
Abstract traits, I should say.
Yeah, I like those a lot because they're kind of in between that like hard and fast genotypic, like this is the gene versus like this is what I can see, taste, touch, whatever. So I like that they're kind of in this in-between place. It's really fun. So anyway, so lentil, the topic now. Tell me about lentil. Tell me about lentil as a crop, the problem you're trying to address in your research. Tell me all about it.
Basically, lentil is a crop from Leguminaceae family, and commonly grown like more than 50 countries around the globe. But we define lentils based on their productions as three major agroecological or macro environment, we would say. Like basically lentils from South Asia and lentils from Mediterranean environment, not the Mediterranean like other regions, but the Mediterranean environment, and also like the lentils in northern temperate, like in Canada or some parts of the States. So basically lentil is one of the major important pulse crop in terms of food security and the nutrition as well. So going forward with the problem that we want to address is in general, like all the breeders, like anywhere in the world, like they want to expand their genetic diversity within their breeding programs. One, basically they want to have like increased yield, which is obviously the farmer's demand for sure. However, like when we make the crosses or introduce new genotypes from different parts of the world, we might face the problem, which is basically the adaptation problem. As for example, like if we take lentils varieties that we grow back home in Nepal and here in Canada, then they might not flower. They flower very poorly, and ultimately will have a very lower yield, which is basically they are not particularly adapted to the Canadian environments, as they exposed to different talent and different temperatures. Likewise, the same rules applies if we take Canadian lentils to Nepal or in South Asia. They might think when to flower, or they might even not to produce, or even they produce flower, the flower might get aborted. So there is some sort of problems going on with adaptations, and we wanted to address that by dissecting some genetics on it.
Yeah. Derek, did you have anything to add?
I was just to kind of give a little bit more history of the lentil crop, and maybe kind of some more background on the adaptation problem that Sandesh was talking about. So the lentils were kind of domesticated in the Fertile Crescent area, and so that's where we're going to have most of the genetic diversity. But then over time, they spread out into South Asia and into Europe, and so now the main production of lentils tends to be in South Asia and in Canada in a temperate region. And so instead of being in their kind of the Fertile Crescent region where there's a lot of genetic diversity, now production is in these kind of extreme ends of the growing environments for lentils, way down in South Asia and then way up north in Canada.
Yeah.
And so that then ties back into that adaptation constraints.
Yeah. I love that this comes up in like anything we talk about that's related to breeding is kind of just balancing the scales on all of these things. I'm such a fan. Yeah, so you completed this GWAS on lentil across different places to help with this problem. So tell me about how you did that. You starting with specifically, you were growing in a lot of different places. So how did you coordinate that?
A lot of collaboration. That's kind of key. So Sandesh and I took care of kind of the growing in Saskatchewan and taking all of the notes here. But obviously we can't do that in the other parts of the world where we did all these field trials. So that, yes, requires a lot of collaboration with other scientists and researchers from other institutions. So basically we send them the seed, they grow it all, collect all that data, and give it back to us. So yeah, the whole collaboration element is very important. Yeah, luckily for us, we had a lot of that. So that was an awesome part of this project, actually.
Yeah, and maybe just one thing to add on is like, as Derek mentioned, it's like huge collaborating kind of projects we carried forward. And this specifically the paper that we are like basing for the discussions today is kind of like a sequel of our previous work, and which we already published, where we talked about how we conducted field trials, like what kind of phenotype or observable data that we collected, and how other sort of things that really triggers the adaptations in lentils are kind of discussed, and this is kind of follow-up things.
Yeah. So how do you find these people to collaborate with? Is it something where you're like, this institution is known for its work in lentils, so I'm just going to shoot them an email? I mean, did you meet these people at conferences? How did you connect with these people?
It would be already sort of the lentil world is kind of already fairly small. So a lot of people do know each other. In the case of, for example, Nepal, maybe Sandesh can kind of expand on that, because Sandesh has a lot of ties to our collaborators within Nepal, and he helped with that a lot.
Yeah, basically, like, as Derek indicated, like one of the like aspects is we go with our personal connections, and also like the project that deals with all these field trials is kind of huge collaborating project itself. So it's not just started for the conducting field trials. It started from the like beginning of when we prepared the project concept notes. So like there's a whole lot of background work has been done on this.
Yeah. Oh man, I'm such a fan of collaboration across the world for science like this. So I find that particularly fascinating. But putting aside my own fascinations with that to move forward, tell me more about like the project itself. So what you were doing, both in that previous project leading into this one. How the GWAS went, how those latent variable phenotypes slotted into all that. Tell me all about it.
So basically the idea was, as kind of Sandesh had mentioned, there are these three major macro environments where lentils are being cultivated. So we took a very large population of diverse lentils. So we had 324 lentils kind of originating from all over the world, and then grew these in all these locations, each of which will have kind of contrasting temperature and photo period. And then collected a number of traits, but our kind of main one was days to flower. So we were really interested in, okay, how does how do these different environments influence when each of these large amount of lentil varieties are flowering?
Days to flower is really important because that triggers when the adaptations occurs, like especially when we take the genotypes from different regions, like that might not produce flower at all, or if they produced, and that might be some temperatures or like the photoperiod or day length difference that might broster flower to die or abort it. So, which is one of the reasons why we are mainly focused on days to flower besides other seasonal events like the emergence or maturity or something. So, and as Derek mentioned, we conducted the GWAS, basically associating or correlating our phenotype data with the genotype data, and we included some new unique approach, which I guess Derek gonna talk later.
So, days to flower, that's considered a phenotypic trait, right?
Yes.
Yes.
Okay. Okay. I was like, where does that fall in the spectrum we've defined? So you were basically trying to grow all of these different varieties in different locations, measure that, and then be able to do this GWAS to kind of tie those traits back to like where it lives in the DNA so that you can know when you are breeding in future, like this has this genetic trait, so we know it's going to do well or not, right?
Yes. Just in addition to that is besides like the regular like days to flower informations and genetic informations, we also incorporated or collected the environmental informations like the day length and the temperature are the main cues here. So it's basically kind of interactions with the environment, phenotypes, and genotypes.
Wow. There's a lot going on there. And then the latent variable phenotypes, you were collecting that. Were you using drones or were you using other data? I know you mentioned it started with kind of drone stuff, but I don't know if that's where that is on this project.
So yeah, the drone stuff, that's where kind of it originally came from, and this will be something that we will be using. However, in our paper where we kind of incorporated these latent variable phenotypes in is, so the kind of the one thing that makes our study a little unique compared, because a lot of people are doing kind of days to flower. However, oftentimes people will be sort of limited to a small number of lines and one single environment, whereas our approach, we had obviously a large population, but then it was by growing them in the multiple environments. That was kind of the key element that allows us to take that our analysis beyond just one line, one environment. We had a lot of lines, a lot of environments. And then as Sandesh, we had the environmental data from each of those environments. And so it was one of the things we were able to do in our previous study was to model days to flower based on temperature and photo period. And so what that did was we were able to get, for each of our 324 lentil varieties, we're able to get an equation to describe when they would flower based on temperature and photo period. So we could plug in, we could take any environment, find the mean temperature and the mean photo period, and then calculate or predict when each of these lentil varieties would flower. Now, in that process of making that equation, we then would get some values that reflect how sensitive that lentil variety is to temperature or photo period. So these were coefficients within the model. But so we were able to, basically extract this value that in a certain way is reflective of the temperature sensitivity or photo period sensitivity. And so that would be one of these latent variable phenotypes where it's just kind of this abstract number that we pulled from an equation that gives us an indication of how, say, temperature sensitive this lentil variety is. And so that's the one of these bizarre latent variable phenotypes. And then so not only that, in addition to having these multiple environments, that's what allowed us to kind of get some of these additional data points. And in this case, because we had these data points that are reflective of environmental cues, we could add that into our analysis, and that kind of helped us quite a lot, because again, it's just going beyond that one variety, one environment. We now can kind of take the analysis to a lot higher of a level.
Wow, that's amazing. Photoperiod is tied to day length, right?
Okay. Yeah.
Cool.
So, yes.
No. I just want to clarify for me. Wow, you guys are amazing. That's so cool. That's so many lines to do that for. I'm so excited. This is great. Hi, everyone. I hope you're enjoying the show. Interested in learning more? Sandesh and Derek's paper, focusing the GWAS lens on days to flower using latent variable phenotypes derived from global multi-environment trials, published in the plant genome, is always freely available. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Thanks again also to our sponsor, Meter Group. Meter specializes in robust soil moisture sensing, innovative weather monitoring, cloud data logging, advanced data visualization software, and more. Their well-published scientific instrumentation is used worldwide in universities, research and testing labs, government agencies, agriculture, and industrial applications. Listen to their new podcast, We Measure the World, to hear how innovative researchers leverage environmental data to make our world a better and more sustainable place at metergroup.com/via-lab-earth. Thank you for being our sponsor. Let's get back to the show. So what did you find then? What were your results?
Okay, so after we performed the GWAS with the days to flower, and in multiple environments, like we are kind of focusing on not just one, but like the diverse environment, we identified some key known flowering genes which are already in there, and some potential new QTLs, which is QTL is basically the reasons, like the part of the DNA where some potential candidate genes are in. And like those QTLs are specific to a specific environment, which is also like kind of new things, kind of like helping the science going forward. So, for example, let's say the implications. I'm going to the implication part. Like, so for example, let's say the breeders in Nepal want to incorporate like Canadian varieties in their breeding programs. If they just make crosses with anything, then they might end up like not finding the best adapted or best suited varieties for their environment. They basically like they'll encounter with that adaptations constraints. But like the knowledge we presented, like the informations of the genes or the marker or which within the DNA, then if they go with that, use that information, they can find or they can cross with the lines that can produce more beneficial lentil varieties in their breeding programs, and that the same thing happens for Canadian situations as well. But having said that, if they end up like something that is not favorable to their environment, they can send back to those lines to Canada, and which might perform well, and we'll also do the same thing for them. So which is kind of like helping each other. Like we are seeing the collaborations in future as well. Yeah.
I love that. Derek, did you have anything to add?
Thanks.
I mean, for our results, like essentially the short answer would be we found a lot of QTL. That would be kind of the short answer. But the longer answer is because we had these multiple environments and we're adding in these latent variable phenotypes, as I mentioned. One, we were able to find QTL that we otherwise would have missed if we were sticking to just a single analysis of just a single environment. And then also we could find QTL that were specific to specific regions. So we can find QTL that are going to be triggered in a temperate environment, so QTLs that will be for temperate environment, and then others that are going to be specific to, say, Mediterranean or South Asian environments. And then also because we were adding in those latent variable phenotypes into our analysis, and in our case, these latent variable phenotypes were our temperature sensitivity and photoperiod sensitivity, we could also use that to kind of get a hint of when we find some of these QTL, okay, is it a temperature related or potentially photoperiod related. So we can also kind of get a hint at what is the potential mechanism for some of these genes. So, for example, in one of the QTL that we got, landed right in a known flowering time gene, and in our analysis with our latent variable phenotype also suggested that this QTL was related to photoperiod. And this because it's a known flowering time gene, it is known to be associated with day length and photoperiod. So this kind of just helped reinforce that, okay, yeah, this is a real QTL, and this gene is the actual causal gene. Whereas in some of our other QTL, one, they didn't land in a gene, so all we know is that, okay, there's just this region of the DNA where we know there's one or more genes related to our trait of interest. And what was kind of cool in our analysis was that we were able to then say, okay, these are probably going to have temperature-related genes in them as well. And that's likely why no one else has ever seen them, because temperature's it's a harder trait to get at, temperature sensitivity, whereas photoperiod sensitivity is a little bit easier, and so that's why some of the already previously known genes are related to that.
Neat. Yeah, I like this a lot. So I have a question. You mentioned that you have like region-specific QTLs. I'm assuming what you mean by that is like they react to the environmental conditions in these specific ones, not that they're like, this lentil has just like an entirely different gene in it than other varieties.
Yeah, correct.
Okay.
Yeah,
just to clarify for myself.
Yeah, so it would be a gene that is influencing days to flower, but only in that environment.
Gotcha.
Whereas if we take it to, say, our environment, okay,
it just doesn't activate
gene,
or whatever.
Yeah,
okay.
Or it's not the main gene, or
Gotcha. Okay. I assumed that was the case, but just to keep things straight in my own brain, I like to ask. So obviously this has a lot of real world applications, both for lentil and possibly other varieties of plant. So can you tell me more about some of the, just the applications for this research?
The modeling approach we talked about on this paper, we in fact we already have like tested whether we could find some genotypes that might perform well in different environments. So let's say if you are breeders from anywhere in the world, and if you provide us the seasonal temperatures, and also the day length informations, and also let us know when you want lentils to flower, and we can fit those informations and say, hey, out of these 324 lines, these might perform well in your particular environment. And now we can also confirm that with the genetic aspects as well from this sort of work. So basically, we're kind of advancing the applications, and which I really hope will be helping. And this is not, I guess, the right time. We are still waiting. But we're in collaborations with some of our partners for applying those technologies, those approaches in the field already. Yeah.
And so to just expand on that, the lentils are grown in Nepal, but there are two other additional regions that potentially, two other new regions within Nepal that where lentils could potentially be grown. So Sandesh was able to use our model to essentially predict when all of our lines would flower in each of those two locations, and then identify which of our 324 lentil varieties would flower at the appropriate time in each of those different regions within Nepal. So if they were wanting to expand lentil production into new regions, that is a kind of, Sandesh was able to kind of give the recommendations of what lentils that they should use.
That's amazing. That's so cool. I love that.
Yeah. And so it kind of has a lot of implications, with climate change and stuff, right? If lentils are going to be pushed into new regions, we can then predict what lentils will flower at the appropriate time. Or like here in Saskatchewan, if we're predicting that, temperatures are going to increase in the future, then we can kind of assess, okay, how is that increase in temperature going to affect when they will all flower?
That's amazing. I love that. Such a fan, spreading lentil, and obviously very important if it's since it is such an important food crop for a lot of people. That's awesome. Yay. Obviously though, there is still more to learn. So what is the future research for lentil breeding and this specific research? Where is it going?
Yes, we are talking about a whole lot of new or unique approaches that we are trying in this particular research. But we are kind of continuing these approaches in lentil itself, like by including the imagery, like the UAV imagery, or also we are also trying to expand the latent variable phenotypes spectrum as well. But besides that, yes, these unique approaches are helping lentils, but these are equally applicable or equally important to test or try on the other crops as well. So we are hoping if someone wants to deal with adaptations, these approaches might be in place to consider for that. And again, like we are doing a whole lot of other things in lentils, like including other aspects as well. Yes.
Awesome. I have 3 questions left for you. The first one is, where can listeners learn more about the things that we've talked about today?
Okay, so within the University of Saskatchewan, we have our one dedicated web platform for like not just the lentils, but whole other legumes or pulse crops. We call that as a no pulse. So if anybody wants to know more about the research or breeding activities going on with the pulse crop, then they can simply go on with the nopulse.usask.ca. And like our supervisor, Dr. Kristen Bates, Professor Dr. Kristen Bates, like she manages a Twitter handles called Wild Lentils. So people also can follow that as well.
Awesome. And that's no as in like knowledge, not as in not a thing. But I will
K-N-O-W.
Yeah. But I will also include links to those in our show notes if people would rather just go there. So then the next question is, if listeners want to get involved in this research more, and that could be as a breeder or a researcher or just as a person that is on the earth, how can they do that?
Yeah, but basically the one approach would be like if they go with the website that I mentioned, like the knowpulse.usask.ca, there is some options for contact us, or we are very friendly people, or very like easy to reach people, so anyone can connect us with emails or with our social handles like the Wild Lentils. Dr. Bate usually like replies, and if she thinks like that should be forwarded to other people, then she usually does that. So yeah, we're very friendly again. Like, so anyone can reach within like like some sort of easy approach, I guess.
Yeah, can confirm. Very friendly people. We'll have your contact information in the show notes also. So if people just want a direct line, they can go ahead and look there. Final question. What is one fun fact that listeners wouldn't know about you if all they had was your research?
I'll start, I guess. For me, I guess it would be just because I had photography as a hobby before I started this. I've kind of become a little bit of an unofficial photographer, full of a lot of stock photos of lentils.
Yeah, like there is high chances, like whenever you see or anybody see photos of our work, there is high chances that might be from Derek.
Nice. What about you, Sandesh?
Like maybe it's not a fun fact, but I also like the fun fact that like we basically in South Asia, we eat lentils at least twice a day. So like eating lentils is not like the big deals for us, and cooking lentils is not a big deal for us. But like whenever I joined this group, like I came here in Canada, like I generally make a soup we call dal, or like I also make some little tweak on my recipes. And our group really loves the final product that I make. So kind of like, so Derek's and my other colleagues or friends called me like, oh, I'm official safe for like making lentil soups for us. So yeah.
Yeah, we decided his soups were so good that, yeah, he's the unofficial chef. And so I'm the unofficial photographer. Sandesh is the unofficial chef.
Ooh, I love that. Yeah, I'm trying to remember if I've like had lentils before, and I'm not sure that I have. So yeah, I guess I need to take a trip to Canada, join the crew.
Well, ironically, lentils aren't super popular here. Like before I joined this group, I had never really eaten lentils. Maybe once or twice my mom had made them, but so I was also in the same position as you. Nowadays, though, I eat them a lot.
Yeah, proximity.
Which tends to happen when you work on a crop.
There you go. You're increasing the genetic diversity to make it more popular, to make more soup. Reach out directly to them and get soup. Awesome. Well, this has been a wonderful conversation. I'm so glad that we got to talk about this research. I'm excited that it's pushing things forward so much. And increasing collaboration, giving ideas to other breeders for how they can improve their crops. So thank you both for all of the hard work that you're putting into your research, both this research and moving forward. And thank you for your time.
No problem.
Thanks so much for the opportunity, like to sharing our knowledge and to sharing what we are doing or what we'll be doing in the future. Thanks so much.
Absolutely. Hi, everyone, and welcome to our Student Spotlight, where we highlight the work of graduate and undergraduate society student members. Today, we'll be talking to Om. Welcome to the show. Can you start us off by introducing yourself and where you're studying?
Thank you, Avi, for this great opportunity to share my research to your audience. I'm Om Prakash Ghimire, second year PhD student in Department of Plant and Environment Science at Clemson University, South Carolina.
Awesome. And what are you currently researching?
I'm currently studying the root system morphology and architecture of diverse cotton plants in the greenhouse and field condition. My goal is to find the beneficial root traits that improve water use efficiency and cotton production, and to identify the molecular markers which are associated with the beneficial root traits, which will be a great resource for cotton improvement and breeding program. For this, I am screening the diverse germplasm of the cotton core set, which represent more than 74% of genetic diversity in whole US upland cotton population. I'm applying non-destructive root phenotyping method in my research to study the root growth and development in the different stages of the cotton throughout the season.
Great. And if you could have your dream research project, what would that be?
That's a great question. I love plant science and especially the root biology and evolutionary biology. As a dream project, I would love to develop the common theoretical framework of the root and shoot system functioning, which can be applied for diverse plants in diverse environment. For this, I would love to study the root system and architecture of hundreds of plant species in different environment and test the common framework of physiology and functioning. This framework will help to understand the climate change effect on plants, evolution of species and the traits, and the crop improvement for climate resilient agriculture.
Awesome. Well, if you'd like to get in touch with Om, we'll have his contact information in our show notes. Thank you so much for being on the show today, and best wishes on your future studies and career. Thank you.
Thank you so much.
Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.